Next, I'd like to invite Professor Tom Maniatis of Columbia University, the director of New York Genome Center, 2012 Lasker Award winner, to give us a perspective of Harvard, Cold Spring Harbor Lab, and Caltech.
Thank you, Sonny. It's an honor and a pleasure to speak today at this extraordinary event celebrating the 70th anniversary of DNA. I'm going to say a few things just about events that were occurring at Cold Spring Harbor Laboratory and Harvard at the time, because that became a very central part of the story. So, in the years immediately after the discovery of the structure of DNA, there was an international effort to understand the biological significance of the structure, starting with efforts by Crick, Brenner, and others at Cambridge, and Seymour Benzer at Caltech to decipher the genetic code using bacteriophage genetics. Shortly after that, messenger RNA was discovered by competitive and collaborative groups of researchers at the MRC in Cambridge, Harvard, and Caltech, and the Pasteur Institute. What I found remarkable about this is that we look back the way we do science now with computers, with email. These advances and interactions were really done by the post, by mail. And so it wasn't as if they could pick up a phone, talk to each other, collaborate. So when you see how they work together and the fundamental advances that they made, it really is extraordinary. So, Jim Watson, this next slide shows what was happening shortly after the discovery of DNA. So at Harvard University, there's Jim Watson, Matt Messelson, who not only was a part of the team who discovered messenger RNA, but also was the one with Frank Stahl that demonstrated the semi-conservative replication of DNA, which is obviously fundamentally important to everything about DNA, including the genetics code and gene expression. After the publication of the structure of DNA in 1953, bacterial and phage genetics and molecular biology were flourishing at Cold Spring Harbor primarily, at the Pasteur Institute. And bacterial conjugation and phage transduction studies provided a detailed genetic map that linked specific genes with function. Earlier, Jacob and Monod and Leboeuf had proposed the extraordinary insightful theories on the regulation of the E. coli lac operon and the lysogenic switch in bacteriophage lambda, proposing the operon theory for which they received the Nobel Prize in 1965. In 1967, Gilbert and Patashny had independently identified and isolated the lac and lambda repressors, the proteins that bind to DNA and regulate gene expression. Both show that the repressors are proteins. They bind to a specific site on DNA. And Patashny went on to work out the extraordinary detail, the mechanism by which phage lambda functions within inside of cells. So these fundamental changes came about, again, because of this community of scientists and how Jim Watson not only discovered with Crick the structure of DNA, but led the science at Harvard and Cold Spring Harbor. So the primary focus of Cold Spring Harbor Lab at the time was yearly meetings of the phage group, which included Jim Watson, Delbruck, Brenner, Renato Del Becco, and others. And Hershey was the first to show that the propagation of phage was a consequence of DNA injection into bacteria. The phage group made use of advances in bacteriophage and bacterial genetics to understand both the genetics and molecular biology of DNA replication and gene expression. They spent extended time in the summers at Cold Spring Harbor Lab and worked closely the rest of the year. This model of international collaboration was extended by Watson to establish Cold Spring Harbor as the center of meetings and courses at the leading edge of biological sciences. A major advance in the establishment of molecular biology was the first edition of Jim Watson's Molecular Biology of the Gene in 1965, which laid out the synthesis of genetics, biochemistry, and structural biology. The book was the first molecular biology textbook, beautifully written and illustrated, and it set a new paradigm in how textbooks were written and illustrated, and led to many other books published by Cold Spring Harbor Press, including the molecular biology of the gene on the left here, and the molecular biology of the cell. Each had many editions and really kept the scientific community updated on all the advances and studies of, and supported the studies of new students. So in addition to Cold Spring Harbor's fundamental contribution to the field, they became a premier publisher of scientific books and a premier convener of international scientific conferences to this day. So I'll finish by noting in 1977 that both Sanger and Maxim Gilbert DNA sequencing methods were published, as Shankar mentioned. Later, Lee Hood and Mike Hunkepeller automated the Sanger sequencing method at Caltech, which made it possible to sequence the human genome, as Shankar mentioned. Most recently, the important paper here by Shankar and his collaborators on arrayed polynucleotides and their use in genome analysis is revolutionized DNA sequencing, as you know. But I meant to mention that, again, this is all about Caltech, Harvard, the MRC, because you see the large-scale automated DNA sequence determination, which used the Sanger technology to sequence the human genome. So these institutions collaborated for many years. They're still collaborating now with the advantages of computers and email and everything else. But I think that the main point I want to make today is that in that moment, immediately after the discovery of the double helix, Watson moved to Harvard, became the chairman of, I think, one of the first molecular biology departments. And it's the interaction between the MRC, Harvard, and Cold Spring Harbor that played a very fundamental role in where we are now in our understanding of molecular biology. Thank you.